Yes, for sure. So welcome back from the break, and now we're happy to have Fredrik Ronquist here. Fredrik is the professor of the Department of Bioinformatics and Genetics at the Swedish Museum for Natural History, and he's also with the Department of Zoology at Stockholm University. Fredrik does research on insect diversity and evolution and on computational phylogenetics. Fredrik has co-developed MrBase that maybe some of you have used, and it's one of the most widely used software packages for Bayesian inference in evolutionary trees. He works with Biodiversity Informatics and acting director for the Swedish Biodiversity Data Infrastructure, and also member of the steering group for the research program in data-driven life science. Fredrik, thank you.
Okay, can you see my presentation now?
Yes, it looks good.
Yeah, okay, that's good. That's good. I thought my task now is to take on my hat as the acting director of the Swedish Biodiversity Data Infrastructure and talk about fair and open science in biodiversity research. And I'm doing this at a very basic level, so assuming no prior knowledge about this field, and just trying to give you an overview of what's going on. Let's see now. Yes, so as a teaser or just an overview of the content, I'm going to talk about how we move international towards fair and open science, and focusing on the two dominant or most important players: Global Biodiversity Information Facility, or GBIF, and Biodiversity Information Standards, and it's known by its acronym TDWG, and I'll explain later why the acronym is TDWG and not something else. Nationally, I'll be focusing on the role of the Swedish GBIF node and research infrastructures supported by the Swedish Research Council, or Vetenskapsrådet, and they have recently merged into a single infrastructure, the Swedish Biodiversity Data Infrastructure, or SBDI. I'm going to talk a little bit about the history and hopefully show that GBIF and TDWG were fair before fair. Since 2010, we're moving towards open science with help from Australia and from other places as well. Come back to that, and finally talk about some things we're working on currently to improve fairness and open science for biodiversity research. So let's start with GBIF, or Global Biodiversity Information Facility. So it's an international network and data infrastructure funded by governments around the world, and it's coordinated through a secretariat in Copenhagen. It's the result of a recommendation from the OECD Mega Science Forum in 1999. This is a quote from the recommendation. It says that it wants to see the formation of a facility that would enable users to navigate and put to use vast quantities of biodiversity information, advancing scientific research, and serving the economic and quality of life interests of society, and providing a basis from which our knowledge of the natural world can grow rapidly and in a manner that avoids duplication of effort and expenditure. So beautiful words. And GBIF was established through an MOU in 2001. Sweden was one of the founding members and is still a major contributor of data, the largest contributor of data per capita. Swedish GBIF node was one of the first national nodes in the GBIF network. It was placed at the Swedish Museum of Natural History, and it's still there. And it's been funded by the Swedish Research Council since the launch. And if you're interested in reading more about this, I provide a URL link there. So in GBIF, since the start in 2001, we've seen 4 data waves. It all started with natural history museum data. And then the data flows or data inputs soon became dominated by citizen science data. So these were data from bird watchers, bird observations, and observations of a lot of other popular organism groups like plants by amateur naturalists. We're now in a phase where monitoring data are becoming more and more important. So these are systematically collected sample or event-based data from, for instance, monitoring programs, long-term inventories with particular focuses using particular methods to collect data. And we're just in the beginnings of the next wave, which is focused more on machine data. So things like DNA sequence data, information extracted from sound, images, video, etc. So was there a question on the chat, or is just that, yeah. Reminder that the chat is open. If I miss some message in the chat that's important, then let me know. We've seen throughout this period an exponential increase of data and usage, and it's been particularly evident the last few years. And what's interesting also, I think, is to note that machine interfaces to the data repositories, for instance, APIs accessed from R, those are increasingly dominating the way in which people are accessing the data. So this is a figure just showing the increase in data records and publications using those data from the entire GBIF network. So in green, you see the number of records in millions. So we're approaching, if we're not already above 2 billion data records for the entire GBIF network. The blue line illustrates the number of peer-reviewed publications using GBIF data, and that those are data per 6 months. So we're now over 2,000 publications, well over 2,000 publications per year using the GBIF data. And just to give you some examples of the Swedish subset of this usage, 10 billion Swedish data records are downloaded from GBIF every month. So that means an average record is downloaded 3 times per day now. In 2018, we went through an exercise where we tried to estimate the value of these services. And we arrived at the number of 2.6 million Swedish kronor in societal savings per day, compared to manual data retrieval for this infrastructure. Of course, truth to be told, a lot of these studies using data through the infrastructure, those would never have been attempted previously because they simply weren't possible to do. So, oops. See if I can go back. So this is just an example of the types of projects we're seeing now. And this is the Lifeplan Project, which is an ERC synergy project. It involves 3 PIs. One of them placed in Uppsala at the Swedish University of Agricultural Sciences, Thomas Roslin. So in this project, they're sampling 200 sites over 6 years and covering the entire globe. They're using a hierarchical sampling design. They're collecting video data, video recordings in the field, sound recordings in the field as well. And they're using DNA sequences of samples, different types of samples. They're using cyclone samplers to collect spores and pollen, using malaise traps to collect arthropods, flying insects to a large extent, and they're collecting soil and root samples, and all of those go through high throughput DNA sequencing. And in total, this project is generating 2 to 5 petabytes of sequence and media data. And of course, when you extract the biodiversity information from that, you get down to smaller data set sizes, but the raw data is also valuable to store, of course. Quite challenging, but this is the future. This is what we're grappling with now. So to make this data available or fair, this organization, Biodiversity Information Standards, has been a key, or TDWG. It's a non-profit organization and community dedicated to developing biodiversity information standards. It was initiated as the Taxonomic Databases Working Group. That's where the TDWG abbreviation comes from. And this was a long time ago, and it happened in the Natural History Museum community. So that's the origin of these efforts. But it's, of course, much broader now. And I think the Natural History Museum group is relatively small in this community now. There are two tasks that this organization focuses on. So one is developing, ratifying, and promoting standards and guidelines for the recording and exchange of data about organisms. So the standards part. But then equally important in developing these standards is that TDWG acts as a forum for discussing all aspects of biodiversity information management through meetings, online discussions, publications, various working groups that work with different standards and guidelines to improve them. To mention example standards, I'm choosing perhaps the most important one here, Darwin Core. It is a glossary of terms, identifiers, labels, and definitions for sharing information about taxon occurrences and related biodiversity information. And it's very widely used. Audubon Core is a more recent standard. It's for sharing multimedia resources and metadata associated with those multimedia resources in particular. There's also a very important standard file format which is used for sharing biodiversity data. It's the Darwin Core archive format. Importantly, various extensions allow addition of new data sources, and they're often incorporated as extensions to Darwin Core. So Event Core is a typical example. It's for sample-based or event-based data from monitoring programs. It allows you to describe the methods used to collect the data, the target organisms, the region that is targeted, etc. And it allows you to collect the information from each sample point. There's also an emerging genomic biodiversity standard developed together with the Genomics Standards Consortium, and Swedes are quite active in the development of this standard. GBIF provides also tools for mobilizing biodiversity data. The most important one, perhaps, is the Internet Publishing Toolkit, which is freely available to data providers. And often instances of the Internet Publishing Toolkit are run at national hubs or regional hubs. Metadata about biodiversity data sets follow the Ecological Metadata Language, or EML, standard. And the GBIF and Living Atlases community provide validation tools based on flagging problems often, but there's some processing also that can happen in some of these validation tools. And I'll come back to the Living Atlases community shortly. If you're interested in reading more about TWG or the Darwin Core or IPT, I provide some links here for you, and EML, which is a separate community. So much of the activities in Sweden in making biodiversity data fair and supporting open biodiversity science is organized through the Swedish biodiversity data infrastructure. It's an informatics infrastructure, or e-infrastructure if you wish, for biodiversity and ecosystems research. It rests on three pillars, or three different activities. It provides convenient and unified access to open biodiversity data. It provides tools for sharing, searching, downloading, visualizing, and analyzing the data. and it provides support for users interested in sharing data or using tools and services for analyzing data. There's in SBDI, there's both a national and international focus. Internationally, of course, the GBIF and TADWIG or TDWG communities are very important. And SBDI includes the Swedish node of GBIF and participates actively in the standardization efforts, but it also participates in several other international initiatives. And nationally, SBDI provides an analysis platform that's focused on Swedish ecosystems primarily. The SBDI technical platform, Living Atlases, is developed collaboratively in an international open source community. The main development happens in Australia, but SBDI and many other partners make important contributions. There are many international unique SBDI assets. For instance, I think it's fair to say we have some cutting edge tools for sharing and analyzing molecular biodiversity data together with other biodiversity data. And we provide access to data from some of the longest running biodiversity monitoring programs in the world. We've been quite active in this area in Sweden since a long time. There's more information on all of this on the SBDI website. The SBDI consortium consists of 11 universities and government agencies, and it's supported by the Swedish Research Council. The SBDI infrastructure is hosted by the Swedish Museum of Natural History. The consortium includes all the main providers of biodiversity data in Sweden. SBDI represents, as I briefly mentioned, it's a merger of many infrastructures. So previously to 2017, a large number of infrastructures in the biodiversity area existed, and many of them were supported by the Swedish Research Council directly by smaller grants. And in 2018 to 2020, these infrastructures coalesced to two initiatives, the Biodiversity Atlas Sweden and the Swedish LifeWatch infrastructures. And from last year, all of this is now under a single hat, SBDI. The Living Atlases community. So this started in 2010 as a national Australian initiative, Atlas of Living Australia, and was backed by mega science scale funding. We're talking about around 300 million Swedish kronor in initial funding for this initiative during the first 5-year period. It was focused from the start on distributed open source software development. And adopted first as a national biodiversity data hub by the GBIF nodes in France, Spain, and later the UK, and Sweden came after that. And now there are 27 regional or national portals deployed around the world, and the community is supported by the GBIF Secretariat, and this community is growing very, very rapidly. So if you want to read more about this, there's a page, GBIF page on the Living Atlases community, and of course you can also see, look at the Atlas of Living Australia, which is the Australian national hub, the first national hub using this technical platform. Yeah, somebody asked me to share the links, but I'm sure we're going to make sure that we share this presentation somehow, so it's conveniently, all the links are conveniently accessible by everybody. So how do we work towards open science within this community? So one thing that's been implemented for a while now is that all GBIF datasets are associated with unique DOIs. So that allows you to cite the data providers, and there's continuous efforts to improve the way that data providers are acknowledged when science is published based on their data. GBIF enforces the use of open Creative Commons licenses throughout for data sets and other aspects of other types of information shared. The GBIF and SPDI downloads, data facets if you wish, they're associated with unique identifiers supporting reproducibility. GBIF also supports publication of data papers in collaboration with Biodiversity Data Journal. This is a quite interesting solution where the metadata can be used to generate draft manuscripts. The Living Atlases community provides an R package for biodiversity analysis using their platform, and that R package is called Gala. SPDI provides an R package, it's called SPDI for R. It wraps Gala and provides additional functionality that are available only for analysis of Swedish ecosystems. We have a lot of additional resources that are not available in other countries. SPDI also provides an online RStudio environment for analysis of biodiversity in ecosystems, and it's called Erosion. And we provide Python-based tools. For publishing and analyzing molecular biodiversity, and those are collected under the name Swedish ASV portal, and ASV is for amplicon sequence variants. And there's a lot more information about these efforts at the SPDI website. Of course, these efforts happening within the SPDI community are supported by many other efforts internationally. And I just wanted to mention one of those. The Open Science community is a very good example of that. And they have focused on different research areas, but biodiversity is one of them. And they have tools for accessing GBIF data, for instance, from R effectively, and for taxonomic annotation, accessing various taxonomic databases, analyzing and resolving problems with taxonomic annotation of biodiversity data. But there's, of course, still lots to do to become fairer and more open. So one thing we're working with in the SPDI consortium is to try to get legacy image datasets associated with Creative Commons based licenses. And that means going back and update previous agreements with users or providers of images, and lots of providers of images. And it's a very challenging task, but we are trying to make progress on that front. One important task we have ahead of us in Sweden is increasing the granularity of large data sets to improve findability and reuse potential. So currently we have a large fraction actually of the Swedish biodiversity data in a large unstructured data set. And there are extremely valuable subsets of that data set where we could provide more, make it easier to find those subsets, and we would also be able to use them more efficiently by providing more appropriate metadata for these subsets of the data. A challenge always is to convince researchers to share the data, make it convenient, emphasize. Many people are not aware of all the benefits now of sharing data. So communicating that, facilitating the sharing, and making sure that the researchers take advantage of these opportunities is still, it's a large part of our work is focused on that. Of course, we want to expand the support for open science with R and Python. One thing we have ahead of us is to provide access to sensitive biodiversity data. So this could be data on, for instance, red-listed species, where sharing detailed information can lead to collectors or people damaging the species that you want to share information about. So this is a quite complicated area, and slightly has some similarities with sensitive data in the life sciences, but there are some unique aspects to it as well. Data cleaning, standards improvement, better publishing and validation tools across the board, working constantly on trying to improve current practices and ways of making data fair and supporting open science in our field. So to summarize what I've covered, we're moving towards fair and open science, I think, internationally in the biodiversity community, thanks to a large extent to efforts by the Global Biodiversity Information Facility and Biodiversity Information Standards Organization. And of course, lots of other players are also important, but these are the key players, I think, if you pick just two of them. Nationally, of course, SBDI and the consortium partners behind it are key players. And we're seeing some emerging collaborations with SciLifeLab Data Centre, DDLS program, NGI sites, and other national research infrastructures that have some overlap, or they provide data or generate data that we want to share and make fair and make it possible for researchers to use in open science in the SBDI platform. So, I don't have time to talk about that, and still in the early days, I guess, in many ways. I think it's interesting to note that GBIF and Tadwig, I think, were fairly early with the type of work that has become known now as fair data. And since 2010, this community has been moving towards open science with the help of this Atlas of Living Australia initiative and the Living Atlases community internationally. And Sweden has been important in pushing the adoption of that standard, that platform across national biodiversity hubs throughout the world. And of course, we're working actively on becoming even fairer and opener. Okay, so that was what I wanted to share. And yeah, I'm open to questions.
Great. Thank you, Fredrik. Do we have any questions we can hear now? I don't see any right away, so I would like to ask you a few things here, because first, one thing that you talk about is that you have data collection is happening for you in that biodiversity. You do a lot of data collection like out in the field. And you also have this component of citizen science. You bring stuff in from bird watchers and people like that. So how do you do that? The part of that concerns metadata and the technical parts to support this kind of distributed data collection. How does that work?
I think in biodiversity data, it's true that we're dealing with many types of data, so these processes need to be tailored to each type of data collection or data flow, I guess. So for citizen science data, it's important to support citizen scientists and making it easy for them to contribute data. You want to emphasize, of course, data quality and data validation, but if you're stressing that too much, then it becomes too complicated for the citizen scientists, and they are less willing to adopt these tools. So one of the SBDI partners, the Swedish Species Information Center, they have a long tradition of working with citizen scientists and developing tools that are useful, that strike the right balance between these different perspectives for citizen scientists. It's just one example. I can take molecular biodiversity data as a different example. There we have a very active group working on that, and they, of course, a lot of the sequencing in Sweden in these projects happen through NGI. And we're trying to, and we're already started, but there's more work to do there in making sure that if you sequence biodiversity samples, environment samples, at NGI that we also make it easy to collect the relevant metadata and publish it through SPDI and share it through the SPDI network together with other biodiversity data. So the answer is different for each data type. I just covered two examples.
That's very interesting. Another thing that I was wondering about, because you've been doing this for so many years, and developing and it's been really impressive to see all the things that you've developed when it comes to data sharing. So about these legacy data sets, what do you see how much are people looking for that? What's the work that you need to put in keeping up quality and for example, like harmonizing metadata towards newer standards and doing annotation? How much of that type of work do you do in order to keep legacy data sets accessible and updated for integration or analysis with more recent tools?
Yeah, that's an excellent question. So I think, in terms of resources, the amount of work that you have to dedicate to legacy data is huge compared to what you can get out of it, and you have to think very carefully about how much it makes sense to work with legacy data. The important thing is, since we're sort of in an exponential phase, increasing amounts of data, it's essential to ensure that the new data comes in a format so you don't have to do the same thing with the new data, because that would be completely impossible. So there needs to be a balance, but I know museums are struggling with digitizing all of their specimen data. Sweden is actually there's a fair number of countries where we've seen large initiatives to digitize natural history collections. And I think Sweden was a little bit unfortunate because some of those initiatives in Sweden happened before we had good high throughput technologies to do this. And since the high throughput technologies, for instance, you can, so a lot of herbaria, you've seen herbarium sheets. So a lot of the plant collections, they consist of largely of herbarium sheets. And there are now automated techniques that allow you to image these herbarium sheets, and you can apply artificial intelligence, machine learning techniques, for instance, to extract the label data from those images, and the images themselves are extremely useful. And you can use the images as a basis for crowdsourcing initiatives and digitizing the data on those herbarium sheets. But the Swedish efforts happened before those techniques were available, so we need another push with mass efforts like that. But it's just, it's a lot of challenging problems, and you have to balance it against making sure that the new data comes in a good format.
Yes, right. Thank you so much. So now on to our next speaker. We have David Diez here. David is a researcher at the Center for Paleogenomics in Sweden, interested in studying the humans and other kind of animal species for population genetics and computational genomics and bioinformatic methods. And the main focus of research now is use of ancient and historical DNA to investigate aspects of intersection between evolutionary genetics and conservation genomics. To you, David.
Thank you. Let me just share my screen. Hopefully you can see the first slide. Yes. Okay.
There's just a couple of black boxes on the slide.
How about now?
Right, now it's perfect. Thank you.
Okay. All right. So the presentation that I'm going to do today is about our newly published pipeline, which is a pipeline, a bioinformatics pipeline to study genomic erosion in endangered species. For what I see, is going to be a little bit of a different presentation style of what other people have been presented today. For starters, I don't have an open word in the title. But I'm going to try to touch some of the topics that are the main theme of this symposium today, and I'll try to do my best. I also don't know very well how to present a pipeline, which is a bioinformatics tool. So I'm going to talk a lot, or mostly, about the topics that made us think about developing this pipeline. So as I said, this pipeline is designed for studying genomic erosion patterns in endangered species. And the key question here is that the wild populations have been seen dramatic declines in population size in the last 200 years. This is a report for 2016. These figures look much worse now in 2022. So the consequence of the population size reductions are species extinctions, which have happened or have increased in the last 200 years as well. In this figure, we have only vertebrates, but the picture doesn't look very good for non-vertebrates either. At the same time, in the last few decades, we have this exponential growth, as Fredrik was mentioning just before, exponential growth in the amount of genetic data or genomes, in this case, that we are being published. Importantly, all these genomes are publicly available, so we can make use of this data for endangered species to look at what's going on with them. Importantly, these genomes are distributed all along the tree of life. So we have reference genomes for things that we never thought that we were going to have a reference genome, and this actually is something that is increasing with time. So we have a plethora of projects, of large consortia or big projects that are aiming at sequencing basically all the living species that we have in the planet. So this is going to solve one of the critical problems that we had when in the conservation genomics field, which is the lack of reference genomes. Obviously, endangered species are not model species. So there was this lack of reference genomes to be able to perform a genomic analysis on this. With these projects now, we have publicly available reference genomes. And so a typical conservation genomics project would look something like this. You would go to the field, sample your endangered species. You would do some DNA extraction, some DNA sequencing, data handling, genome analysis, and of course, once you have all the results, then you can go and apply some conservation measures based on those data that you just obtained. Unfortunately, the reality is much more complicated, and so obviously, the path from genome data to conservation measures is not so straight, and it's actually quite more complicated. So it looks more like a path like this, and the reasons are multiple. And I'm not going to go into these questions, but I want to focus on at least two of them that are relevant for this symposium, which is the lack of reproducibility between studies and the standardization of measures. And this is relevant for endangered species when we want to compare what's going on with different species, and we want to compare them at the genome level. So yes, this is important, and this is among the objectives that we had when we decided to develop our pipeline GeneRoad. Now, when populations are growing small, then they are increasing the risk of getting extinct. The reasons for this are twofold, and they have to do with stochastic, demographic, and environmental events, things that can happen to the species just because the population is very small. Imagine that you have a population in an island, and then there's a hurricane, so all the individuals can die. If you have that species in different islands, then a hurricane may not get this species extinct. This is a stochastic event that can happen if your population is very small. Another process that can happen that can lead to extinctions are genetic processes, and these are related to the size of the population. So theory predicts that if you have a very small population, then they can enter into this so-called extinction vortex. So imagine that you have a small population already because of whatever process that happened before, for example, human hunting. And then this they enter into this extinction vortex. It has, simplifying a little bit, it has two different sides. It has the inbreeding depression side, in which the small population is causing the individuals to mate with close relatives, which isn't going to increase the inbreeding. This inbreeding is going to unmask deleterious alleles, which in turn are going to reduce the fitness of the population, causing inbreeding depression, and so the population is going to grow smaller, and so it goes again into the cycle. Genomic evidence for this are reduced heterozygosity and increased inbreeding, and you can estimate these values from a single genome of an individual of this population. The other side of the extinction vortex will be the mutational meltdown side, in which a small population is subjected to a strong genetic drift just because of the size of the population, which means that the predefined selection is relaxed. So natural selection cannot get rid of the very deleterious mutations just because of the genetic drift is so strong, so it overcomes the strength of the selection. So this leads to an accumulation of deleterious alleles or deleterious mutations that are actually reducing the fitness of the population, and so the population goes again into this cycle of being smaller. Genomic evidences for this in the genomes of an endangered species, that will be an increase of number of deleterious mutations in the genome. So with these ideas in mind, back in 2018, we thought that it would be a good idea to compare different species and see how some of these indices actually compare with the level of threat that these species have in different lists. So we were looking at the red list of IUCN, and then we compared the levels of genetic diversity, one of the indices that I was mentioning for the extinction vortex, and we were comparing those with the level of endangered. So you have critical endangered, vulnerable, all these concerns. So it goes from more endangered to less endangered. And we see here in this figure that even though the lower the genetic diversity, the population size is also lower, which is something that we would expect. This is not related to the status in the red list, so it's not immediately clear how these indices are reflected in the red list status of the species. And prioritizing species in the red list is extremely important for conservation, because that's mostly going to govern how the money that we have for conservation is going to be invested. Which species are we going to be injecting some money, and which ones are we going to be injecting less? The reasons for this connection between genomic diversity and red list status seem to be related to demographic history events on each species, and also life history traits and some others that I'm not mentioning here. But the conclusion is that if we want to prioritize species, we cannot just do it by comparing levels of genomic diversity in modern samples, because these are poor indices, poor proxies, for conservation status. So then what do we do? So back in 2018, we had this suggestion in which, instead of only sequencing modern samples from endangered species, what we should be doing was should be sequencing also samples from before the bottlenecks happen in this species. So okay, here we have an example. This is the Sumatran rhinos, a species that is critically endangered. There's about only 30 individuals left in the wild. And then here we have a model of how the demography of an endangered species would look like today. So in here we have the ancestral population size, that would be NC0, which was big, large. At some point, there is something that happens to this species, usually humans, causing a strong bottleneck, and then this is time. So in here, today we have a very small population. This would be the resulting size of the population. And instead of only sequencing individuals here, which is what the conservation genomics field was doing until now, our suggestion was to also sequence individuals back when the population was large, and obviously you can find those individuals in museum collections if there is vertebrates, and sometimes you are lucky if the species that you are interested in study is well represented in museum collections. So back to the example with the Sumatran rhinoceros, we have, this is in light green, we have the historical distribution, which was quite large in 3 islands, Sumatran, Borneo, and the Malay Peninsula. And then in dark green, we have only the remaining populations or the areas of the remaining populations, which is basically only in Sumatran and Borneo. So the idea would be to sequence from samples that are from the original range and then samples from the modern range and then compare those. So in the minimal example, you would sequence 1 genome from this historical population back when the population was large, 1 individual from the modern population now that the population is very small, and then instead of reporting just the values, you would report the what we call the delta index, which in this case, it will be just subtracting the value of the historical sample, using it as a baseline, basically. Now, if you do that, instead of just providing 1 value, you're informing about the rate of change in these genomic erosion indices. Imagine that you are just estimating heterozygosity. Here, instead of giving just 1 value, you're giving a rate of change in heterozygosity through time, which is much more informative of what's happening with this species than just a static value. And in turn, we think that this actually helps to provide comparable patterns or comparable trends that you can use to compare different species. So instead of providing 1 value that is tied to these demographic events that happen and they are different between modern species, what you're giving to the public or to the community is a value of the trend that is happening to that population or to that species. And we think this is much more valuable for prioritizing conservation efforts in endangered species, such as for the red list. So this was, again, one of the objectives of us developing the Genome pipeline. Okay, so now that we have all the objectives, then I'm gonna talk a little bit about the pipeline itself. So this is a pipeline that which main aim will be to simultaneously analyze genome resequencing data from ancient or historical and as well as modern samples. And the aim is to look for patterns of genome erosion by comparing the historical samples or the ancient samples with the modern ones, as I just explained with the example of the Sumatran rhinos. The pipeline is developed in SnakeMake, and we have worked hard. So there is no programming and knowledge required to run this pipeline, only certain knowledge of the command line to be used. It does use Conda and Singularity containers, so it's highly portable. You can use it in basically in any cluster. So it's designed for high performance computing clusters because the amount of information that it processes is quite large, and also the space that you need is quite large. But depending on the number of samples that you have and the purpose of your analysis, then you may be able to run it in a computer. We also wanted to make it as modular as we could. So there are different phases of the pipeline that you can run individually. And we always say that it's highly flexible because the amount of customization that you can do when you run the pipeline is also quite large. And it's also very well documented. So we have this wiki in the GitHub pipeline that is quite extensive, and it goes from background information on the methods that we use or that are used in the pipeline to information about installation and also most common problems that we have found while developing the pipeline. And there is also an article that is in preprint state, so it's already published. So these are the objectives that we wanted to achieve when we develop the pipeline, or some things that we wanted the pipeline to achieve when we were developing. And so one of the main objectives was to make the analysis that we do in this pipeline to make them as reproducible as possible, so people can reanalyze the data and obtain the same results using the same settings. So this can also lead to an increase in the standardization of indices, and I'm referring here to these genomic indices. I'm going to explain a little bit about those in a second. But standardization is very important for what I was mentioning before, just to be able to compare between different studies. And also standardization is also achieved within the pipeline, because the main objective of the pipeline is to compare samples from different time points, so historical or ancient and modern samples. So that's one of the main objectives. And we want to believe that it's accessible to non-experts, so you don't need to be a bioinformatician to run the pipeline. So it will be accessible for general public or people that are no that have no programming skills, for example. So I can go very quickly through the main parts of the pipeline, and then so in the minimal amount of data that you need to run the pipeline, you need a reference genome, which is good because basically all the species are going to have a reference genome very soon. You may have an annotation or not. We do have analysis that are involving annotations, but we also have alternatives that work without annotations, so it's not an absolute requirement. What you do need is samples from 2 different time points, so ideally historical or ancient, depends on the species that you're going to study, on the problem that you want to tackle. And then you also need modern samples. In the minimal example, you can use 1 sample from each, so 1 historical, one modern if you want. So we have this data processing track first. As I said, the steps are modular, so you can run up to any of the steps that you want, but they all have to be run in succession, so you cannot just do one of them. But basically, you start with FASTQ data, basically from the sequencer, and then you are going to map the data to the reference genome. And after a few processes, you will get a bound file that is comparable basically between the historical samples and the modern samples. And of course, the nature of the data of historical and modern samples is different, so they are treated slightly differently, but the pipeline is aware of this, so they should be comparable in the end. There are a few additional steps, especially for the historical or ancient data. Depends on if the data is treated for damage or not, and if you're interested in the mitochondrial data. And we sometimes use this to estimate levels of contamination in these samples. In the second part of the data processing step, we have the genotyping, which is performed for both the historical and modern samples. And then we have these GBIF scores data processing as well. This is the part of the pipeline that can be that accounts for not having an annotation for the species, for the reference genome. So I will explain in a second how we use the GBIF scores and why don't we need the annotation. So then we have the data analysis track of the pipeline, in which we actually produce output results from our sequencing data. And again, we have different tracks for the historical or ancient samples and for the modern samples. In this case, because the data was processed being aware that there is historical or modern data, then they should be comparable at this point. But importantly, every individual is analyzed separately, so they are not biased by the data from other time periods. Downstream results that you will get from our pipeline would be, for example, you get a PCA principal component analysis, which you can check for looking for sequencing errors and other stuff, population structure between historical and modern samples and so on. You do get estimations of the levels of genomic diversity and inbreeding per individual. And we do this basically looking at a number of heterozygote sites that you have in the genome, and also FRO, which is the total amount of the genome that is compressed in runs of homozygosity. So the fraction of the genome that is allocated in this runs of homozygosity. And then we have two ways to estimate mutational load. So basically accumulation of deleterious mutations when you compare 2 time points, in this case, historical and modern. We have 2 methods. One of them, there will be the method based on SNPF and the annotation for the reference genome. And then we have another method that is based on GBIF scores. And that is not depending on the annotation. So this is when the reference genome doesn't have annotation, you can use these GBIF scores that are based on the alignment of several reference genomes from other species. It's a panel of species, or a panel of reference genomes, and then you don't need this annotation using the GBIF score. So the outputs of the pipeline, apart from a wide range of quality control files that you get of basically after all the steps, you do get a full report in which you get a summary of the steps that have been run using the pipeline. You get principal component analysis results for all your samples plot together. You do get levels of heterozygosity for your samples. In this case, imagine that your samples, historical samples, are yellow and your modern samples are blue. So this is how it will look like. You get a report for the level of inbreeding that you have per sample. It's based on this FRO that I was mentioning before. And then you get 2 possible estimations of mutational load, one of them based on the annotation and the other one based on these GBIF scores. So I just wanted to give a little example on the kind of studies that you can use using this pipeline. So in this study that we just published last year in the Sumatran rhinoceros, most of the analysis were actually run using an in development version of this pipeline. So as I mentioned before, the Sumatran rhinos is a critically endangered species, and in this study we did sequence a few samples from the modern population that is remaining, but we also, from 3 populations, so there's the 2 remaining ones, Sumatra and Borneo. We also sequenced some individuals from the now extinct population in the Malay Peninsula. And we also had samples, historical samples from at least 2 of these populations, the Malay Peninsula and Borneo. So the idea was to use this setup to compare levels of changing levels of genetic diversity using our pipeline. This is an example of how the results look like. So here we have the FRO, which is basically inbreeding levels, and then we have for each population, Borneo and Malay Peninsula, we have 2 different bars, one of the historical samples, and then you have the modern samples. So the idea is that you can compare this between the different populations and the different time points. And here we can see, for example, that the historical populations in both Borneo and the Malay Peninsula had lower levels of inbreeding than the modern populations. In this case, the Malay Peninsula is already extinct, but they have had higher inbreeding. Relative mutational load would be the GBIF scores that I was mentioning before, and it doesn't seem to do it. At least there is differences in the Malay Peninsula, so the historical sample had higher mutational load than the modern population. There are no significant differences in Borneo. This is also because there's only 1 sample in the historical pool. And if you look at another measure of deleterious mutations, that would be the number of loss of function variants in each individual, you will see that the Borneo historical population had lower number of loss of function mutations than the modern ones, and this is what the theory was predicting. So as soon as your population grows smaller, then it should be accumulating deleterious mutations. This was not what we were seeing in the Malay Peninsula, though. So this is just an example of what kind of studies can be done in using this pipeline, and then here I have examples of other species, endangered species or not, that we have been using the pipeline with to study these patterns of genomic erosion. So the pipeline is publicly available in the GitHub of the MBIS, and yeah, that's
thanks. Yeah, well,
yes.
thanks, David. That was one of the questions right away too. Was if you have links to your resources, and I'll ask because Fredrik's on still too. But if you could share links to your slides, both of you, as well, so we can put that in our materials. So that's the first question. We don't necessarily have enough time, maybe, but briefly, I guess one question is, I guess the challenges for sharing, sharing this information, and some of the positives, if you could quickly share that, and then we'll move on to our next speaker.
Sorry, I didn't understand. What do you mean?
Oh, well, so some of this, it seems like it's come up before, the sensitive information, like sharing some of the sensitive information versus the highly collaborative nature of your project too.
Mm.
So how you work around both.
Well, I don't know if I understand the question. You mean sharing our information with the genomic data, or?
Yeah. Because the population, right? Sensitive
Right.
data, and yeah.
Right. So for the genomic data is always public, as in basically in all these and this in our studies, basically we publish the data and it's publicly available. Anyone can use it, but there is sensitive information when it comes to species such as the Sumatran rhinos, for example, where you sometimes have geographical location data for individuals that are very endangered, and then they're subjected to poaching, for example. So those information are usually hidden in our studies, and then sometimes we do have the information, sometimes we don't even have the information, just because of the people that we are collaborating with, they refuse to give it away. So I know that this is an interesting topic for people working in conservation projects, especially with big mammals, which are the ones that are usually very, very few left. But yeah, the genetic data is usually always public. Metadata sometimes is not.
Well, thanks. So if we have any follow-up questions, we'll, if people can ask them in the chat, but thank you. We're on to our next speaker, who is Yohan Rao. And I'll just briefly introduce Yohan. I've had the pleasure of working with Yohan a lot lately. He's the research scientist with Cooperative Institute for Satellite and Earth System Studies at North Carolina State University. His research focuses on using long-term satellite data and machine learning for climate studies, and he has a passion for open science. He's actually working with us on a number of open science projects. So Yohan, if you're there and you can share your slides, and also share a link if you have that in the chat. So we've been also trying to share the slides with everyone too. But thanks, Yohan. Welcome.
Thanks, Chris. Can you see my screen okay?
Yeah, we can see it. It's a bird picture.
Yes. Right. Hi, everyone. Like Chris said, myself, I am a research scientist at North Carolina State University and working with several data centers here on climate and environmental research. And here, my title is something called "If It's Broken, Why Fix It?" That's something we heard a lot when we're trying to make some changes to all those existing pipelines. So I want to share some of the open science lessons learned when dealing with those questions and some experience I had. And So a little bit more about myself. I come from background. I was trained as a statistician, and then I find my way somehow into earth and environmental sciences. So this satellite image can probably capture the past 80 years of my work, that looking at satellite data and how we can use satellite data to improve our understanding of climates. And this is a satellite captured by NOAA 20, which is a polar optical satellites, and overseeing study area called Tibetan Plateau. Not, I assume that you know this, but it's one of the third highest, well, we call it third pole of the world because the high elevation of that area. So for my dissertation research, I look at how to use satellite data and in situ measurements to improve our understanding of the climate change in that area, because we have really poor data records in those like really not populated area. Because when we are set up station to measure temperature and the climates, those are usually in those well-populated area because that's how the weather station started. And so this quote on this image basically is something I wrote in my dissertation. So that I want to dedicate my dissertation to everyone who is committed to promote open data, open source projects, and open scholarly research, just because without them, my research would probably be impossible, just like the satellite data when they are not open, like in the 80s and 90s, that would take months, if not years, to get those satellite images, not let alone to get those long-term records of satellite image for the 30 to 40 years. So I consider myself being on conquest to on the open science, just because that although open science has been mentioned so many times, that not everyone is still fully on board the open science. So for myself, I am at this triangle area. Okay, by the way, the image I took from Chicago about two months ago when I was there for a conference. And on the right side, I use this triangle to define myself, that I really enjoy doing the field work. That's one of the reasons where I switched from statistics to earth and space science, environmental science, because I really enjoy going out to do the field work. So I know how hard and how difficult collecting data can be. And so that's one of the reasons I want to touch on later, that when people are mentioning that I spend a lot of time to collect those data, what I should share with others before I can even publish my results. And then the satellite is another big motivation that promoted me to get into the open science, just because I see the value of those really open satellite imagery can produce for the society. And myself as amateur coder and a program developer, trying to bridge those field data as well as satellite data for some application research. And I benefited tremendously from those open source code and tools and softwares that everyone has been developing and sharing on websites. So I see that myself has been shaped by a lot of open science efforts others have been doing. So I have trying to contribute back for the open science topic. So here are some lessons learned, well, not lessons learned, some questions or some of the statements that I often get from all this phenomena observed in the past, maybe just 2 or 3 years when I get started doing some more projects with in the more operational world, when people have been doing similar things for like 2 decades and how to change people's behavior. So the first thing that I noticed is hello world meets email firewall. I will talk more about this email firewall. I recently have been encountering it a couple of times. And the second argument is that we collected data, why we should make it open. There's always, or why I should not get any specific recognition in becoming a co-author or something. There's always the ... that people have different ways to argue why they don't want to share the data. And the last is what the more to the topic, that we have always been doing this that way. And if it ain't broken, why fix it? So let's go into the first one that I want to talk about, is the email firewall. So when we started to learn coding, that the first example we always use is the hello world. That means that you're connected to the internet. And then through the internet, that now when we start sharing data, sharing our research, that email have been really common to get those requests of the data and code. What left side is an example that I got just like 3 months ago that as regarding one of my research I did during my master's. That's about 9 years ago, I think. I'm thrilled to have somebody like reaching out to me saying, hey, I really enjoy reading your work, and this method looks really interesting. I want to use it to compare with my research. I was like, that's fantastic. But then that brought me to think, oh, I'm doing the same thing. So I have been setting up an email firewall to allow people to access the research I've been doing in the past. To be fair, that's 9 or 10 years ago. If I'm doing it, I will probably not do the same thing when I'm publishing my research right now, but that's 10 years ago. And I want to emphasize in the open science, it's always a learning journey for many, even for people who have been in the open science for a really long time, that can, sometimes we still need to change and improve. So that this is one of the anecdote story I have, and I've been trying to be more open with like sharing my code on GitHub directly and put them into the report as well as in the publication. So, and another story I want to share is that just happened a month ago. So that I submitted research to one of the journals that it's a high impact journal, and that the reviewer came back with that, oh, you may need at this data set G. Let's use the initial of that data set G, not using the full full name of data set for revision. I was like, that's fair, because that data set has been really popular, has been around for like 2 decades, if not more. And so on day 3, I started search, oh, where I can find this data. So of course, they always point you to those original publication from the 2013 or the improved version that's from 2013. It's like, okay, I want to see if I can find data statements, availability statements, nothing. And go to the PI's website. So I started to go into request the data from the PI. That's my second failed attempt. That's like 10 days after I got my review back, and I really need to get on going with getting those data. And that started making me start to panic, just because it's already, I tried all different ways trying to find people who may have access to the data, trying to get the data for my revision, for my research. And almost like 3 weeks later, I basically just throwing darts on the board like blind. So requesting data from one of the PI's former postdocs. I was like, hey, I see you use this research data set a couple of times. So by any chance, you still have this data? And he really generously shared the data using Google Drive. And the Google Drive was created on 2017. And the data set had been claimed to keep being updated. So at this point, I basically just throw my hands up. It's like, yeah, I probably just use this data and trying to meet the requirement, because I really don't have enough time to trying to chasing down this data set even more. So I started drafting the review and then adding a sassy comment that like, despite my best effort, I only able to get the data from 2017 for this data set. So hope this satisfy the revision of this requirement. So this is another like story. I really hurt myself, and that's why I'm really passionate about trying to make the data as open as possible. So that's why some of those like, community guidelines produced, such as American Geophysical Union, where Chris has been working with others, the late editor-in-chief of one of the AGU journals, Peter Fox, has been one of the champions on this, is the data and software sharing guideline for authors submitting to AGU journals. But I want to emphasize in one thing, that we all have a role to play, either as authors, reviewers, managers, developers, and users, whatever role you have to play to improve the data and software sharing through this guideline. And so I've been reviewing for some of the AGU journals and have been really, I would not say strict, but really strongly encourage people to follow the guidelines. So when I see people not sharing their results or sharing their data, I was like, hey, this is a community guideline. You should really follow this. And so there's always a back and forth between myself, editor, and the authors. And sometimes I get small victory. I got really excited. I will share with Chris, even I'm not sharing the name or the work, but just sharing with Chris, say, hey, I finally get somebody to share even the secondary data, not necessarily the raw data, because usage rights. But it's a small step towards success. So, and I found this to be kind of like irony. So when we are young, or when we were taught by the parents, or when we're teaching our kids, is that sharing is caring. But then as researchers, somehow we sort of forgetting this like golden rule that we have been taught, or we have been teaching others, that sharing is caring and data is ours, because you can always find different reasons, say the data is ours and I can share, but certain things need to be met. This some of the requirements are really important, because there are some of the usage rights. For example, sensitive information you don't want to share, that's totally legit reason, but there are some reasons that can be a little bit on the edge. But once I get into open science and then data sharing, and navigating data sharing can be a really interesting question, just because we want to decide what we want to share and how we want to share it, and who decides what data should be shared and what data counts, especially when the ethics comes into play. That's the data ethics can be a really interesting topic that we have been thinking about ethics and data ethics on some of the applications like environmental application, as well as the data science, artificial intelligence. And one thing that I've been really interested in learning more is the care principle when it comes to the indigenous data, the governance, and the ownership of those data. So especially when we are developing some applications that may have potential impact on those underrepresented communities, or those communities who often don't have a say in the data, how the data is collected. One thing I've been thinking a lot about is that I use satellite data a lot, and now with more and more high resolution commercial satellite data become available, and you can easily observe the tribal lands. And when you're doing any applications or research, how does the ownership of those data count? And if you're doing any resource monitoring, that becomes an ethic concern for the data users and data producers. But looking on the bright side, that we always have some improvement and progress. One thing that IPCC Intergovernmental Panel on Climate Change has been really excited to see this is their interactive atlas from the Working Group 1, looking at physical science basis of the climate change. This interactive atlas is basically a website that you can go to choose what data set and what different climate variables you want to look at, and for what specific region. So if you go there, that you can just basically have those open information readily available for any users who want to go there. They also have open access to all their frameworks. So if you go to the github.com/the IPCC account, Working Group 1, that you can find this atlas. They produce all those codes and share those codes and the data sources, so you can reproduce the results. So this not only open science can increase our access to the data, but also increase trust, so become transparent and reproducible for all those entire workflow that has been the backbone for those climate negotiation and also climate research. So that leads me sort of thinking inwards about my work, because I work at the National Environmental Data Center. So we have tremendous amount of the environmental data that can be used. But now we are actually looking more inwards toward who the data we're serving to. That we have really diverse user for Earth environmental data science data, because those Earth environmental data from different sources, from satellites, from those like power measurements, from citizen scientists, and just those camera captured by those stations. And we're also getting into genomics, omics data from the ecosystem. So we have a tremendous amount of data that can be a treasure to answer a lot of the societal challenges like climate change, like environmental equity and justice. And we have really diverse users, and how we can make the open science. I use a quote from UNESCO when they are recommending the open science for the entire community, is that accessible to all and benefits to all. So that leads me and some of the researchers started thinking how we can increase accessibility to our environmental data through open science. That with open science, we have tremendous tools can be leveraged to access those information. And the IPCC Atlas is a really interesting example. And I'm actually following this steps, trying to increasing the usability and discoverability of some of the data, the long-term satellite data records, and more information I will be sharing in the summer through a website that we will launch our project website for that. So one, going back to the topic or the themes, the title of my talk is that if it ain't broke, don't fix it. I always go to XKCD to find those inspiration, comics to use. So this two are all from XKCD. One is that every time when you're updating something in the existing workflow, there's always those long-time users saying, hey, the updates broke my work or broke something on my end. So can you just reverse it back? Because we have been doing it a long time that way that any updates can just screw things up. So let's don't do it. And then on the right side, it's more ironic that oftentimes we are fixing things, and the longer you spend on fixing it, not always that goes to a really high return. Sometimes you get into some of the failures, but it's okay. But when you finish the updating or fixing things, that you can get really things working great. So I just want to use this to start this conversation that we are having here. That one of the project I started 3 years ago, I joined 3 years ago. I don't want to use the exact name of the data or project just because we are improving. So I don't want to naming any names here, but like the project started in 2001. And the whole workflow starts from the raw data all the way to the final product. It goes through several stages. So in the first stage, the first release of the product in 2007, then second stage in 2011, and then last stage, as somewhat we are improving upon, is 2016. And for the entire workflow, that we have 4 different languages, trying to go from Fortune. At that time, they're still using Fortune 77, not even Fortune, the newer version of Fortune. And then we're using C in the middle, and then MATLAB come into play, and the IDEA come into play. So the entire workflow can be complex. I'm not saying that it's really wrong, but just like when we, as a researcher, when we started doing our own work, we tend to pick the tools that's mostly readily available that we're familiar with. And there are several personnel change in the project. So the people's preferred language started changing. So the entire workflow becomes, I call it workflow stew, just like you put everything in it and hope for the best of the results. And I started reading more and more about the technical debt in the software development. So it all becomes the trade-off between the long-term, short-term convenience versus long-term sustainability. That using this comic example, that when you have been doing the same thing for like a decade or so, that you are using the triangle shaped wheel, that you think that get the work done, and why you want to change things, and you are getting too busy. But when the term of technical debt in software development is that when we are choosing to the short-term convenience, it's okay, but then we always need to pay the debts in between. We basically front-loading all those comfortness that we have, and to do the development, make things work fast. But for the long-term goal, we really want to think about sustainability. We need to repay the debts, trying to change the code and make it more sustainable in the development process, and can be easy to use in the future and maintain in the future as well. So that's when we started reconciling technical debts. That it doesn't cost me a lot to convince people to get on board, just because they know that it should be changed, really. And so that we started using Python and R as the main framework for us. And you might ask, why you still use 2 languages? Just because R is good at doing certain things, and I am a native R person. So Python is another. They're good at 2 different things. But a great thing is that in the R community, there's something, or as well as in the Python community, there's something called Articulates that is trying to integrate R and Python. And in the Python, there's a similar package, integrate R into Python. So you can always easily integrate this too, especially with some of the interactive tools like the notebook Jupyter, that you can easily have those Python that are running the same documents. That makes it much easier. But I think another thing I want to emphasize is that the process requires really strong version control that you need to start doing. That I have the horror story of talking about this project from version 3 to version 5 to version 2016, and then going back to another version number. That's not most efficient way to do version control. GitHub, Git exists for a reason. And there's always a constant dialogue with those project teams and the stakeholders to think about how you want to make open science work for your team. And documentation. I have been burned by so many times for documentation topic that I was trying to go through a fixed coefficient that have been used in the workflow, and there's no documentation about that. And so it took us about a week or two to figure out 11 line of the coefficients in that code and what that means and how is that exactly correct or not. So that documentation is really important. And I think my time is about to up. So I'm gonna this is also my last slides. You can always find me at my email or Twitter or through Chris and others. Thank you so much.
Thanks, Yohan. You're right on time, actually. So we didn't get any questions in the chat or our Q&A just yet, but I did want to start with the usual one, which is I don't know if you shared them earlier, the link to your slide deck, but I think you're going to do that right now because you had some really great slides there as well. Yeah, I guess like the other one, maybe you can start as others are maybe asking questions, but I think you really bring up something really critical, which I think is the challenge that everyone sort of comes to us at some point, is the technical debt, the effort that it takes, right, to make these transition to being, to adopting some of these new technologies, being more open, being more efficient. One of my favorite papers out there is Our Path to Better Science by Julia Stewart-Lowndes, who really kind of documents this. And I guess like where I'm driving at is that you've been spending all this time to work through some of that technical debt because you believe in it. But I also wonder about sort of the reward side of it, and sort of doing this work. Where have you seen the benefits, with regards to sharing more of your work more openly and making it more accessible? Would love to hear your work on the sort of usability side that you were talking about too. But I don't know if you can speak to that, to the other side of like, the sort of recognition, the rewards, and other side. Sorry, big question.
Yeah, I mean, reward is always a harder thing to measure, especially when we're talking about open science. I think especially in research world, that paper is at least currently is a gold currency, right? And open science can be a second level, but now I see a lot of people changing it with the National Academy of Science within the United States. They're talking about incentives. And one thing that I think is building the community, I found a tremendous community by doing this open science, that if you doing open science, if you're on social media, just on Twitter, just simply tweet, and then you can get a response like in I would not say 5 seconds, but 5 minutes, that there are all those open science people online trying to answer questions in those ecosystem. And that's sometimes those responses are even faster than my project team, that when I was trying to go through email and trying to find those legacy coefficients or those legacy issues in the codes, that we need to schedule a meeting to talk about it. And on making the progress on project, sometimes it's even faster on open science side. That for example, PanGeo is a great geoscience community using Python, where if you have any questions to accessing those open earth science data on PanGeo or those core packages, just tweet or go on the community platform, that you can find all those great resources and people answer your questions that help you to go through. So I think that's one of the biggest rewards you can find is the community. And the other one is the recognition unofficially, because officially there's still things need to be changed, but unofficially that people are recognizing your work and then reusing some of your code and work. And I saw some most random recognition of my friends because of some of the code they have been working on and sharing online and in some AGU conferences. So I was like, oh, I know that person in the acknowledgement section because we used to work together really closely. And I always take a picture and send to my friends, hey, your code has been used by others. And then here's just an example of the great work you have been doing. So this is something I can share.
Okay, so we had one question. It was for the paper, right? Our Path to Better Science link, and I guess you're still in the process of. Maybe you can give us a preview of the work that you're doing. I guess it's in GitHub, right? The usability work that you were talking about, the accessibility of your.
It would be a website like inspired by the IPCC Atlas. So it would be a part. It's a visualization and customization portal built on website that you can easily create a global map of the some climate data records online without downloading anything, and we'll produce a notebook. If you want to access the data from the clouds and do analysis, there's a notebook teaching you how to do it.
Well, great. So we're on time again. Thanks a lot, Yohan. This brings me to a point where it's I can encourage the people that are attending to contribute to our Etherpad. We were just talking about links to helpful resources, and our Etherpad. We're trying to get some helpful resources linked to there, and also maybe particular questions that you'd like to see answered in sort of an open science training program, open science curriculum. So, some of the things that you mentioned, Yohan, and also previous presenters as well. So if you have time, please go into the Etherpad and really share anything that you think is useful there. And I will share the link if someone hasn't done it yet in chat yet. But we also are at a break. So we have about 10 minutes before we come back again. And Daniel Bangert will be with us to talk about his work in Ireland around open science. So if you need to stretch your legs and to ponder open science some more, or get a hot beverage, then this is your time. So please just take a break, and we'll be back in about 10 minutes. So thanks again, everyone. And we'll pause. Unfortunately, we don't have waiting music here, but if you wanted to listen to that, and feel free to